Assalamualaikum. Hope you all are fine. So in today's lecture, we will be discussing about the R and S configuration of the amino acids. In our previous lectures, we've discussed two basic enantiomers of the amino acids, that is L and D forms of the amino acids. And as L and D forms of the amino acids are directly linked with the Fisher projections of the amino acids, so in other terms, you can say that the Fisher projections are used to represent the L and D enantiomers of amino acids. So we also briefly discussed about the Fisher projections in our previous lecture, but we can have detailed analysis of the Fisher projections in our this lecture as well. But to keep it in mind that L and D configurations, which we discussed in our previous lectures, these two enantiomers or these two forms of the amino acids can be represented by the Fisher projections. So first of all, you have to know what are basically the Fisher projections. And secondly, in today's lecture, when we are going to discuss the R and S configuration of the amino acids, R and S form of the amino acids, these are also the two forms in which an amino acid can exist. We will discuss these two forms of the amino acids with respect to the Fisher projections, plus with respect to the wedge projections as well. So these are the two projections which are used to represent the R and S configuration. For example, you can be given an amino acid in the Fisher projection form in your exam, or you can be given an amino acid in the wedge projection form. These are the two representations in which amino acids can be drawn or can be written. So, and you are asked a question that just show that this amino acid is in the R configuration or it is in the S configuration. So first of all, you should know what is actually the Fisher projections and what are actually the wedge projections. So let's briefly discuss the Fisher projections first. Fisher projections are a kind of the projections which actually show that amino acids exist in the tetrahedral symmetry. So in order to represent the amino acids in the tetrahedral symmetry, Fisher projections are meant to highlight, they actually highlight or they actually show the position are actually the presence of the four important functional groups, with respect to the chiral center of the amino acid. And you all know what is basically the chiral center of the amino acid. Those who are basically new, I should encourage you to basically see my previous lectures to determine the chirality of the amino acids and just have a brief look into it. This is basically the chiral carbon or the chiral center of the amino acids. Why it is called as chiral carbon? Because this carbon at one time is attached to the four different groups, 1, 2, 3 and 4 different groups are attached to this chiral center, and that is the alpha carbon or the chiral carbon. According to the Fisher projections, the highly oxidized group, that is actually this group, carboxylic group, is given the top position, just like it is present in the top in the chain in case of the sugars. So it is given a top position in with respect to the chiral center in the structure of the amino acids. And similarly, the R functional group, which is actually variable, it could be different for every type of the amino acid. For example, it could be a ring structure, it could be a chain structure, it could exist in the form of an atom, in the molecule, as an ion. So as it is variable, so it is given in all the amino acids given almost the bottom position. But I'm talking about this in case of the Fisher projection. So in the Fisher projection, it is given the bottom position. So two positions are actually clear. Now let's talk about this second group and the fourth group, which is amino group and the hydrogen group. And the Fisher projections, these two groups should be in line with each other, mean in the horizontal line. This is basically the amino group and this is the hydrogen atom, and they should be in front of each other. So these are given the side positions from the chiral center. So this is all about the Fisher projections. So in case of the Fisher projections, there are actually basically the two vertical bonds here, and there are two horizontal bonds here, and Fisher projections represent the symmetry of these bonds with respect to this chiral center, that how these bonds are arranged or organized with respect to the chiral center. And according to this symmetry, the amino acids basically exist in the tetrahedral form or tetrahedral structure form. And Now let's move towards the wedge projections. This is totally a new kind of the projection which we are going to discuss in our this lecture. So according to the wedge projection, again the chiral carbon is given the center position definitely, and it is a chiral one because it is attached to the four different groups at one time. Now here you can see the arrangement of the bonds is bit different here. So first of all, keep it in your mind that Fisher projections represent the bonds position with respect to the plane. So according to it, it actually differentiate the functional groups based upon their position with respect to the plane. And first of all, the first rule that the Fisher projections basically follow is the wedge projections basically follow that particular type of the functional group which is actually present behind the plane, mean when you are viewing the structure of the amino acid in a 3D form, and when the amino acid, now just let's suppose that this structure is in front of you, and when it is in front of you, that hydrogen is actually, this hydrogen, this functional group is actually present at the back of the structure, so it is not clearly visible to you. Let's think this in the, let's basically suppose this thing in your mind that amino acid structure is in front of you and hydrogen functional group is behind that structure, behind the molecule, and you cannot actually mean clearly you cannot locate, so it is just hiding behind the structure. So that any particular functional group which is hiding behind the structure, or it is behind the plane, that is away from the viewer, that is basically represented in the dotted line, just like that. It is also called as wedge hatch line as well. So next one rule is clear. Now the second rule is basically that that particular type of the functional group which is just in front of the plane and towards the viewer, that is much towards the viewer, mean amino group is just in front of you, mean projecting towards you, and it is basically you can easily look into that group, and it is in front of the structure. That particular type of the functional group is represented by these, by you can say the dark solid line. So these are all solid line, this is solid line, this is solid line, this is solid line, but you can say this is a dark type of dark solid line, so represented by a dark solid line. Okay, now we are left with the two groups, that is this group which is basically the R chain actually, in case of basically the alanine, because here we are also looking at the L-alanine structure, because amino group is at the left side of the chiral center, so whenever it is at the left side of the chiral center, it is L form of the amino acids, and we have discussed this in our previous lectures as well. And we are left with this R group and this carboxylic group, this is the fourth. Okay, so why they are represented by the normal solid lines, those structures which are represented by the normal solid lines, why? Because both of these groups are in the same plane, they are neither projecting exactly towards the viewer, or they are not exactly away from the viewer, they are basically in the same plane, in the medium plane, so we basically represent them in the normal solid lines. So these are both represented by the normal solid lines. So this is how you basically represent the amino acids according to the wedge projections, and this is the wedge projection of the L-alanine. Now let's look at the wedge projection structure of the same amino acid, that is alanine, but in the D form. And again, you know that in the D form, the amino group will be projected on the left side of the chiral center. But here again, you can see the amino group, as this is the type of the functional group in the wedge projection, that is towards the viewer, out of the plane but towards the viewer. You can clearly see this particular structure represented by the high solid lines, or you can say the dark solid line. And again, the hydrogen functional group, which is basically on the back side of the structure, that is away from the viewer, or you can say it is behind the plane, that is represented by this kind of the dotted line. And similarly again, these two groups, that is R group, R methyl group, and similarly the carboxyl group, which are in the same plane, they are basically represented by the normal solid lines. So I have already discussed this. So this is basically the structure of the D-alanine in the wedge projections form, and this is the structure of the L-alanine in the wedge projections form. And similarly, this is how the L-alanine is represented in the Fisher projections. And again, you know that in the D-alanine form, the amino group will be on this side. There will be the amino group, and there will be basically the hydrogen. And we have discussed this thing so many times in our previous lectures. So now, if you are given an amino acids, whether in the Fisher projection form, whether in the wedge projection form, you could be asked that now you are going to tell that whether this amino acid is in the R configuration and whether it is in the S configuration. Okay, I have erased the stuff from the L-alanine and that of the D-alanine, so we can basically solve these two amino acids which are present in the wedge projections to determine whether these amino acids are in the R configuration or in the S configuration. And in order to determine the amino acid whether in the R and S form, we will be basically following a particular type of the system of rules, and that is called as CIP, which basically stands for con, inglot, prelog, priority system. And what this system actually mean for? This system actually represent that whether a particular type of the molecule, whether it's an amino acid, whether it's basically a protein structure, whether it's a DNA molecule as well, whether this particular type of the molecule is in the left-handed orientation or in the right-handed orientation. And again, left-handed orientation basically means the molecule could be in the anti-clockwise rotation or orientation, and similarly right-handed means the molecule will be in the clockwise orientation. So according to the R and S configuration, the R basically stands for rectus, and rectus in the Latin words basically means right. So whenever an amino acid is in the clockwise orientation, when our amino acid will be in the clockwise orientation, it is basically in the right-handed form, and we will call them as R is in the R configuration. So I hope this is clear. I'm again repeating this, that whether if our amino acid is in the clockwise orientation, it means our amino acid is in the right-handed form and it is in the R configuration. And similarly, if our amino acid is in the left-handed form, it is oriented in the anti-clockwise direction. So it will be in the S configuration, because in the Latin word S stands for sinister, and sinister basically means left. So our amino acid will be in the S configuration. So I hope this is basically clear to all of you. Okay. Now let's have a basically let's try to basically solve these two amino acids, whether they are in the R and S configuration. So just keep an eye on the cursor. Second thing, okay, just let me complete this CIP system. The second thing which just this CIP system actually indicates that it actually prioritizes the atoms on the basis of their atomic number. So it actually prioritizes atoms. And how it prioritizes the atoms? It prioritizes the atoms based upon their atomic numbers. And the atom which basically has the highest atomic number, it is given the priority 1. And the atom which has the least basically the atomic number, it would be written in the last. So let's do it. Okay, in case of the amino acids, we are basically dealing with the mostly 4 to 5 different types of the atoms. So in case of the 20 different types of the amino acids, we mostly deal with in the proteins. They usually have the 5 different types of the atoms, like they have the sulfur, and they can have nitrogen, oxygen, carbon, and hydrogen, most importantly. Now how we are going to basically prioritize them? We are going to prioritize them based upon their atomic numbers. So let's prioritize them. Now by considering the CIP rule, we will be solving the wedge projections for both L-alanine and the D-alanine, because we are going to determine that whether these two amino acids are in the R configuration or in the S configuration. So first thing you have to do in order to solve these two amino acids for the RS configuration, you have to point out those atoms which are directly attached to this chiral center. So in case of the methyl group, this is actually the carbon which is directly attached to the chiral center, and in case of the amino group, this functional group, this is a nitrogen atom which is directly attached to the central chiral carbon, and in this case this is obviously a single atom which is directly attached to the chiral center. And in case of the carboxylate group, let me open this structure. We have this structure in this open form, and again in this condition, this is actually the carbon which is directly attached to this chiral center. So once you have identified all those atoms which are actually directly attached to the chiral center, now you have to give the priority numbers based upon the rules which are set by the CIP system. The table is in front of you, and from the table you can easily identify that sulfur is given the highest priority based upon its atomic number. Then the second priority is given to oxygen, then there's nitrogen, and then there's a carbon, and then there is hydrogen. So we have identified the atoms which are directly attached to the carbon. Let me point them again. This is nitrogen, carbon, hydrogen, and here is the carbon. Okay, now we are going to allot them the priority number. So based upon these atoms which we are now considering in the alanine, we are dealing with the nitrogen, carbon, and that of the hydrogen atom. There is no sulfur atom. So among these atoms, now look at this table. The nitrogen is given the highest priority because we are dealing with nitrogen, carbon, and hydrogen, and among these three atoms the nitrogen is given the highest priority, so we will give it the number 1. And among these three, definitely hydrogen has the least atomic number, so it will be given the priority number 4. Now we are left with the functional groups, that is this R group and this carboxylate group. And the problem here is that in both these cases, this is the carbon atom which is directly attached to the central chiral carbon. Now what to do in that particular case? In that case, you are going to look at the second atom beside the carbon atom. So it is, in this case, it is oxygen, and here in this case it is basically the hydrogen. So among the oxygen and hydrogen, you know that it is actually the oxygen which is given the highest priority. So oxygen is given the highest priority as compared to that of the hydrogen. So second number will be given to this functional group, and definitely the third number will be given to the methyl group here. So now we are done with allotting the priority numbers to these 4 functional groups which are attached to the chiral center. Now the second CIP rule is that now when you are done with the priority numbers, the second rule is that you have to count these numbers and you have to move around the amino acid in this form, that you have to read from 1, 2, 3 via 2. This is very important. Now you are going to read from 1, 2, 3 via 2. Why CIP rule has ignored the 4th number, that is basically the hydrogen, because this is actually in the dotted form, and dotted line actually indicates that this hydrogen is actually on the back side of the structure, behind the structure. So it doesn't come in the orientation while determining the clockwise orientation or anti-clockwise orientation of the amino acids. We are considering only those 3 functional groups which are in front of the viewer, which actually is determining the orientation, whether clockwise or anti-clockwise orientation of the amino acid. As hydrogen is backside of the structure, away from the viewer, so it is generally ignored. So now what you have to do, you have to move according to the CIP rule system. You have to move from 1, just follow my cursor, 1, 2, 3. You have to move from 1 to 3 via 2, means you have to pass the 2 and you have to move towards the 3. So now in that case, let me erase this stuff, and I'm going to draw this again for a better understanding, that Again, we have numbers. Like we have given this amino group number 1, 4, this is second, and this one is third. Okay. Now, what you have to do, you have to move from 1 to 3 via 2. Okay. And now you will be looking at the arrowhead, that at which point this arrowhead are in which direction it is actually moving. It either it is moving in the right direction or in the left direction. So in that case, it is moving in the left hand direction. And according to the CIP rule, if it is moving in the left hand direction, this molecule is oriented in the anti-clockwise orientation. So it has anti-clockwise orientation in this case. And when there is an anti-clockwise orientation in the RS configuration, it will be considered as sinister form. Or sinister means you know that this means left, so it will be considered as S configuration for that particular amino acid. Okay, so I hope this is clear to you. So first of all, you have to identify the atoms which are directly attached to the central chiral carbon. And then what you have to do is that you have to give priority numbers based upon the atomic numbers. So one with the highest atomic number will be given the priority number 1, and one with that of the least atomic number will be given the priority number 4. And then you have to move from 1 to 3, ignoring 4 via 2. And then you have to determine that whether the arrowhead is moving in the left-handed direction or in the right-handed direction. And then based upon this, you have to determine the orientation of the amino acid, that whether it is anti-clockwise orientation or clockwise orientation. If it is anti-clockwise orientation, then it will be the S configuration of the amino acids in the RS system. Okay, and you will be writing your answer that this amino acid, which is actually in the LS system, and it is L-alanine. When we solve this amino acid according to the CIP system, this amino acid will exist in the S configuration in the RS system. Okay, now let's solve the same amino acid but in the D form, because in this case the amino group is in the right hand direction, and you know that when it is on the right side according to the chiral center, it is the D form of the amino acid. Now again, now in this case you know that which atom is directly attached to the chiral center. Just move towards the second step, that is to allot the priority numbers. So now we are going to allot the priority numbers, and you know that nitrogen is given the priority number 1 according to the CIP rules. And among these, as you know that the hydrogen has the least atomic number, so it will be given the priority number 4. Now we are left with these two functional groups, and again we have the carbon. So we will be looking at the center second atom, that is hydrogen in this case, and this is the oxygen in this case. So definitely the priority will be given to oxygen, so it will be allotted with the number 2, and it will be given the number 3. So again, according to the CIP rules, you have to move from 1, 2, 3 via 2, ignoring 4. Okay, because it is at the back side of the molecule. So now you have to look at the arrowhead, that is which direction it is actually moving. Now it is moving in the right hand direction, and when it is moving in the right hand direction, it means that it is oriented in the clockwise form. So molecule is in the clockwise orientation, and when it is in the clockwise orientation according to the CIP rule, it is actually in the R configuration, rectus configuration. So it has R configuration. It is R configured. Okay. So I hope this is clear to you, that how you are going to determine that whether an amino acid is in the RS configuration when you have been given the amino acid in the wedge projections. Okay. In the next part of this lecture, we will be solving structure of different amino acids present in wedge projections and Fisher projections to determine whether they are in the R configuration or S configuration.